Well, I am Frank Muller-Karger from the University of South Florida. The University of South Florida is located in Tampa. We have a campus in St. Petersburg, which is on the Gulf of Mexico side of Florida. And it's not quite South Florida, it's actually kind of in the middle of Florida. And you're all welcome to come. One of the things that we're trying to do, and you'll see, and I'll repeat it several times, is we're trying to develop a network of people that are interested in seeing how life in the sea is changing. Okay? And so the question is, how do we do that? How we're trying to really focus on the Americas right now as a first place to start, but we want to do this globally. So we want to do this linking Africa, Asia, Europe, and Australia, and so on. So we want to develop a way that we can compare measurements over a long term to see really how we can determine whether life in the sea is changing and then how that affects people. Okay, so this is a very important concept about this Marine Biodiversity Observation Network that we call MBON. And a lot of the talk that I'm going to focus on is on the structure of this MBON, what it means, how we're trying to do it. And so it's a little bit of a bureaucratic talk as opposed to more of a science talk. There's going to be some examples of what we're doing from a science point of view, but I was asked to provide an idea of these international frameworks for how people do ocean observation. So you have my email up there, and you're welcome. There's 3 of my students in the audience. There's Cara, Savannah, and Luis Liscano way in the back. And I've asked Cara, Savannah, and Luis to work with you and try to identify people that are interested in sharing data, and especially if you're trying to measure biodiversity and living things on shores. We're starting to, with very, very simple protocols that are very inexpensive to do measurements on beaches and in the intertidal rocky shores. We're working with the Smithsonian and the Intergovernmental Oceanographic Commission on this, to see if we can then collect data in the cheapest way possible to then make some informed assessment of how life is changing. Anyway, so I guess we're going to miss a little bit of the screen. But these are some of the questions that are fundamental in marine ecology today. Okay, so I'm going to read my own slides here. If everybody hears that we're losing biodiversity. Okay. How many people have heard this here? Everybody. Do you think that's true? How do we know that that's true? Can we actually measure? Can we actually say that? Oh, thank you. No? That's okay. We just missed one letter. That's okay. The way that we are making that statement is a little bit you know, it takes a lot of hubris. I don't know if you know the word hubris. It takes a lot of courage to say something like that when we don't really know. So my concern is that we're making statements about the world without really being fully informed. And the reason that people are saying this, that we're losing biodiversity, is an assumption, because we're developing the coast, we're using resources, and we're effectively shrinking habitat for some species. So the assumption is that biodiversity is being lost. And yet, and I'm going to touch on this several different times, is when you say biodiversity is being lost, it's a dangerous statement, because sometimes it depends on how you measure biodiversity. Biodiversity may be going up, but it may be going up in a way you don't want. Like you may have more types of bacteria in a place, so biodiversity went up. But it may be a bunch of pathogens, and so and you're losing species that you want to conserve for your own benefit. So we're very interested in the sustainable development of societies around the world. A lot of focus on this, and yet people are talking about things in a way that is not very clear, especially not very clear to the general public. So all of this is an effort to clarify things for the scientists, to develop some best practices and some simple methods that then can be told to people, and especially decision makers, on how to address some of these. So other questions. How does biodiversity affect productivity? Okay, there's a lot of evidence that the more diverse a system, the more productive it is. But is that true? I mean, how do we test this? This is not very easy to test. There's a guy at the Smithsonian, Emmett Duffy, that has published several papers on this, because traditionally, when you think about ecological principles that I learned when I went to the university, it was the dogma in ecology was that if you had fewer species, like in temperate zones or in upwelling areas, you could have more productivity. But it turns out that in many cases, when people can measure this very carefully, and you define diversity very carefully, it turns out that the more diverse systems are more productive. So this is a science that is not settled yet. There's a lot of discussion and a lot of experimentation that has to happen. How does biodiversity affect water cycles and water quality, and vice versa? Okay, we don't really know that, and this is different everywhere. Is coastal biodiversity related to what happens on land? And so we're really pushing the idea in many of these circles that I'm going to talk to you about, that we need to bring the land community, so terrestrial scientists, together with ocean scientists. And as you know, there's been kind of an unfortunate divergence between limnologists and oceanography. It turns out that 50 years ago, 100 years ago, limnologists and oceanographers worked very close together. Now they're completely different communities, and they meet differently. They have different talks. They publish in different journals. And so we're trying to bring that back together. But we're also trying to bring together the terrestrial community and the ocean community, because we talk about the same thing. We talk about biodiversity, we talk about productivity, we talk about ecosystem services, and in the end, it's one Earth. It's not 15 different Earths, the way that we have divided the scientific community. So the way that we have divided ourselves is a little bit artificial, and we need to bring that back together. So how does all of this affect society? Can we quantify the value of biodiversity? And that's not easy. Yesterday, Dan Suman said that it's easier to measure people than biology. Well, I don't believe that. I think it's very, very difficult to do anything in terms of indicators that make any sense and that is useful. Because how do you come up with an indicator, and what is an indicator is something that I'm going to talk about, and what makes an indicator useful. So in the end, is how do we measure diversity in the ocean, and how do we track it over time? And it's not easy. Even today, after 200 years of oceanography, there's very little that we can say, because the data sets that we have are very poor. And I'll show you the data sets that we're trying to use. We're trying to encourage people to share information. Maybe you don't need to share everything that you do, but if we want to understand how life in the sea is changing, people are going to have to share data, and they have to share data in protocols that are common, that follow best practices, that have documented metadata. And if we don't do that, we'll never understand the ocean. So it takes a lot of energy to share your information, to be open with your data. But what you'll find is that once you share data, you will become parts of groups and people that invite you to publish more, not less. There may be one or another person that may steal your data. It always happens. But it will open doors. So if you share your information, you'll gain. You will not lose. Anyway, this slide is kind of a summary of the entire 3-hour talk. So if you fall asleep, this is what you have to remember. We're trying to come up together with this MBON concept, the Marine Biodiversity Observation Network. That's part of a larger effort called Geo Bond. It's a group on Earth observations. The group on Earth observations is actually a consortium, a partnership between over 100 governments and NGOs and private organizations. And the idea is to, within this group on Earth observations, is to make Earth observing data, being satellites and in situ data, available to make decisions. So, and there's other parallel efforts to do that. So we're now trying to we're trying to organize thinking so that these different governments that have formed these international bureaucracies coming back together again. So networking is the whole concept. We want to document best practices, so we're establishing and trying to promote the idea that we can use the United Nations as a place to put best practices, document it, curate it, make it very openly available. And then the idea is to make all of this in an operational sense. So there's a lot of basic science that we do that we don't call mission-oriented or operational. Operational means that we're going to do things on a routine basis, supported in a sustainable way to provide information to people for making decisions. Okay, so operational is a very key word that is not just one time or one time for research. Okay. Is everybody following me? Am I talking too fast? If you have questions, please raise your hand, stop me. Sometimes I go too fast, and I want to make sure that everybody understands what I'm saying. So I'm going to also talk about the framework for ocean observing. The framework for ocean observing is something that came up under the auspices of UNESCO, and it's promoting something called the Global Ocean Observing System, or GOOS. And there's many different regional organizations that participate in making of GOOS. I'm going to talk about what we call essential variables. This business of essential variables is now a big deal internationally. It's linked to the Sustainable Development Goals, and there's now everybody kind of realized that if I call something essential, maybe I get money, and we need to be careful about that, because the danger with that is that we'll have an explosion of things that we call essential, and then nothing is essential, okay? And it becomes really, it works against us. There's two of these. Of course, there's essential climate variables that you may have heard of. This is where all this started about 10, 15 years ago. And that's what the Intergovernmental Panel for Climate Change uses. So they track indicators like global temperature, okay, for that's an essential climate variable. And then from that, the ocean community started developing essential variables of its own. So we have essential ocean variables, and I'll explain what those are. And then we have essential biodiversity variables on the other hand. And so those started to develop completely separately, and we need to bring them back together so we don't have this crazy divergence in people competing to do just one variable and not another one. So those are the key words: essential variables, the Global Ocean Observing System, the GOOS, and then we have the Geo Bond. So these are there's a lot of acronyms over here, but so we'll try to explain them as we go along. So these are things that you already know, but I'll go over it anyway, because it's the way that we explain things to people when we try to have people understand the importance of the ocean. Most people don't think about the ocean. They're worried about life every day. So not only does the ocean cover over 70% of the planet, it's on the average 4 kilometers deep. So all of this is living space. There's life throughout this space. It's not just at the surface, and yet it's very, very difficult for us to measure what happens below the surface. And one of the issues that we have with measuring how many fish there are, how many other creatures there are, is getting below the surface. Very expensive, very tough, and the ocean is very big. Life is also involved in changing the planet. So not only do we have exchange of gases, oxygen, carbon dioxide, these kinds of things, there's other kinds of gases that are involved in a life cycle, but we have a lot of geology. So reefs and the depositing of silica, calcium carbonate, and we have other types of forms of life that change the geology of the planet, as well as the biogeochemistry or the chemistry of the planet. Then we have a great diversity of life, but life is also, this diversity of life is important for how we live on this planet. So, and one of the problems that we have right now is we all have oversimplified life on Earth. So we measure carbon. Everybody talks about carbon credits. Well, if we all were the same, just one piece of carbon, it wouldn't be that interesting. And life is, this diversity of life is what makes this planet work. It's not just one type of life. The diversity of life is critical to sustain life on the planet. And yet we don't think about it. We just talk about carbon, or we just talk about nitrogen. And so we have oversimplified things to a point where it's very difficult to track what really life on Earth is doing. And so we have artesian fisheries or recreation. We have pharmaceuticals, we have transportation, we have fisheries. All of these things are related in one way or another to life. And what we're trying to come up with is our strategies to conserve life in some balance of the right type of life that is beneficial to us, but also develop. We know there's going to be another billion people on this planet in the next 20 years. So how can we ensure that we don't completely screw up the planet so that we end up with more wars and more poverty? And then that's going to be very, very difficult, okay? And that's why we talk about developing the ocean. It's not just conserve everything or lock it all up like somebody suggested yesterday. We wish we could do that, but it's not realistic. We're people, we eat, we throw away things. So we need to manage the way that we do this in a sensible way, in a realistic way. We still want to make money, we want to have a good life, so it's not realistic to lock away everything so that nobody else can use it, only us. That's not a good way to look at things. And yet we have an ocean that is almost completely unexplored, and so we need to develop the technology, the science to be able to do that, and then use these resources in a way that we don't completely use them up too quickly. So there's many different organizations around the world. World Bank, this thing called the OECD. There's many different groups that are trying to put value on how life in the ocean, or the ocean in general, is leading us to this blue economy. So there's lots of buzzwords. And blue economy is anything that has to do with the ocean that makes money, hopefully. And in this case, for example, there's biology that is throughout. It's not only fisheries, but in terms of tourism, the aquaculture. Many of the oil and gas are related to biology in the way that you do ecosystem assessments and so on. So many different aspects of the maritime industry or the blue economy is related to biology, and so we need to keep track of that. And the expectation is that this ocean economy is going to grow by several $billion to almost $1 trillion over the next several decades. And yet we talk about this business that the planet is changing in a very negative way, and so we need to be careful in that we measure how the planet is changing in the proper way. So even now we go to children's books in school, and it says, well, human activity can reduce biodiversity in a children's book. Okay, so so they don't get, children will not understand what this means, and I can tell you that most people in this room really have no idea whether this statement is true or not. Okay? And yet we're saying it. We're saying that we're introducing species, we have invasive species everywhere, so we need to think about exactly what that means. And in fact, yes, the planet is changing, and the question is how, and how do species move around? Is this a natural thing? Is it an artificial thing? So we have different studies. Of course, we can measure the temperature of the planet. We know it's changing. We can measure oxygen in the water, although it's very difficult to say that the entire ocean is now becoming hypoxic, like some people are saying. So we need to be careful in really understanding the way that we can measure hypoxic regions in the world and whether they're expanding or not, or if it's just a movement of water. There's still controversy about that. We have harmful algae blooms. A lot of statements saying, we have more harmful algae blooms around the world now than we did 30 years ago. Is this true? Or are we just measuring better? Are we taking more measurements? Okay, so today, a lot of these statements don't really have a good scientific basis, and yet we're doing it, we're making them in a way to get money for science. And so we need to be careful that we don't end up with a scary situation, but we also want to have a realistic, real, validated science that can help inform people about what is really happening. There are studies like this. This is Ben Halpern. He published this a few years back that show the footprint of what humans have done in the world's oceans. There's almost really no place in the world that we have not in some way impacted. Okay, so humans have a big impact on the planet, and yet whenever we talk about ecology, humans are not included. Okay, so we need to change that thinking. People are part of the ecosystem, and we need to bring people back into the ecosystem because we do change things. We have this building where there was no building before. We have cities where there were cities before. We change the planet in a big way, so we have to understand that impact. So, life in the sea is changing. Biodiversity is tied to ecosystem production in many different ways. We want to manage the ocean better, although it's really hard to say that we manage the ocean. We really manage people. We manage people's behaviors. And so there's a big, whenever we say ecosystem management, we're not managing the ecosystem. We're managing the interaction of people with our ecosystem. So that's a very important point. But we need information. And so that's where these observing systems come up, is we're trying to organize, there's a big efforts to try to coordinate what we measure, how we, so if we want to measure the temperature of the planet, how can we gather the information? Is that information good? Are the satellites that are measuring this properly calibrated? How can we compare information from Europe to South America? Are the thermometers well calibrated? Well, thermometers are the simplest technology that we can use to do this now. But there's other things. We want to measure pH, we want to measure nitrogen, we want to measure carbon, and yet in carbon dioxide, and you have no idea how difficult it is to make a pH measurement in the ocean. And the scales change continuously because people that do this for a living are changing the indicators that you use to measure pH in a way that actually jumps more than what we think pH is changing. So it's very difficult to have a realistic understanding of what is happening with the world right now beyond temperature changes. We want to come up with technologies that are cheap, that you can deploy in different places, that can measure life in the ocean in a better way. Right now, there's a lot of technologies in different brands of technology that measure fluorescence. And do you know who measures fluorescence here in the ocean? Nobody? Do you know can you explain what that is? Yeah, so fluorescence is a measure of phytoplankton productivity and biomass. If you take a plant, any green plant that has chlorophyll, phytoplankton or plants over here, and you shine blue light, that chlorophyll will emit red light. That's called fluorescence. So plants will take on blue light, they will absorb that in their photosystem apparatus, and in that chlorophyll will then pump some electrons out to a longer wavelength, and that will be emitted as red light. We have devices that we call fluorometers that shine blue light on algae, the phytoplankton, and then you can measure the red light. And that's a measure of biomass. The more chlorophyll, the bigger the signal. But it's also a measure of productivity because if you don't saturate the chlorophyll, it actually tells you a little bit about the health of the algae or the health of the plant. So right now, a lot of people are going around, they're buying fluorometers, putting it on a mooring or putting it on a profiler, and they're saying, okay, I'm measuring biology because I'm measuring fluorescence. It's a physical measurement, it's not a biological measurement because it's light. And it's very, very difficult to understand a measure of fluorescence because the plant will fluoresce differently depending on whether it's healthy, not healthy, depending on the species, and depending on if it's day or night. And it's very difficult. So it's a good measure. It's a very simple measure, very common today, but it's difficult to interpret, and it doesn't tell you anything about biodiversity. It doesn't. It's a bulk measure like carbon. So we need to go beyond fluorescence. And how do you do that? You need to have cameras, microscopes that you can put in the water, and these things exist. So there's there's a flow cytometer that you can put in the water right now. You could put it on a mooring, or you can put it on a cable, or you can put it on a boat and put water through it as a flow-through system. Yet the device costs $100,000. You can have a little camera that we call an underwater plankton a video recorder, or a UPV, and this thing can profile in a buoy. It can be put on a CTD and go up and down on a wire. And again, that little camera costs $100,000. So how can we get better measures of biodiversity using these types of devices if they cost $100,000? It's not realistic. So it's only a few groups around the world can sustain or maintain these types of devices. So we need to bring the cost of technology down and distribute it more broadly to try to understand that. We also need to find better protocols for measuring things in situ by people. So that's a thing that I'll talk a little bit about that. And then try to understand how these measurements are useful for society, which is not easy at all. Okay, so with MBON, what we're trying to do is we're trying to come up with a global community of practice that focuses on these issues, that comes up with common methods, protocols, that shares at least some data that we can compare, and that we can communicate the importance of life in the sea to other people, to the public in general. So MBON is a program of Geo Bond, and I'm going to explain what Geo Bond is in the context of these essential biodiversity variables. So this is a diagram that was published as part of the framework for ocean observing almost 10 years ago. The whole concept here is that you have groups of variables that you can collect that don't measure everything. They're essential variables that help you explain climate, that help you explain something about the ocean, they help you explain something about the land and something about the atmosphere. In the case of the ocean, we have the essential ocean variables, and I'll tell you where that comes from. And then we have the essential biodiversity variables that come up under a separate branch of bureaucracies. Okay, so we're trying to look at the intersection that help us explain life in the ocean. And this is the nightmare that we have with the international governance systems that we're creating. And on the one side, I'm going to talk about first on your right-hand side, that's GEO, that's a group on Earth observations. And on the left-hand side, we have the United Nations. They're completely separate right now. They're trying to find links. On the left-hand side, we have a lot of ocean activities under UNESCO, and that's the group under UNESCO that is trying to organize, is actually organizing ocean observing, is a global ocean observing system under the Intergovernmental Oceanographic Commission, and there's several different programs within the IOC, Intergovernmental Oceanographic Commission, that tries to do that. And on the GEO side, we have all kinds of different programs that try to share in situ and satellite data, as well as biodiversity data. And again, these are the same governments of 100+ countries around the world, yet the representatives from each country that goes to the GEO meetings are different people that go to the UNESCO meetings. And so often they don't talk to each other, and so there's a disconnect. So we're trying to make these connections at a lower level, at least at the science level, so that when we float them up through the plenary at the IOC or through the conference of the parties of GEO, that there's some sense. You know, the United Nations has many ocean-relevant programs, like the Convention on Biological Diversity, the UN Convention of the Law of the Sea. Many of these things are, you have UN Environment, your FAO. So all of these things are not necessarily well connected, even though they live under something called UN, and even though they're made up by the same governments around the world. So I'm going to talk about the GEO side first. The Group on Earth Observations was established in 2005. And the whole idea was to create this global Earth observing system of systems. Okay, so you have a climate observing system, you have a terrestrial observing system, you have an ocean observing system. And the whole point was to try to unite these things under a common framework. And they're doing this, and this is actually happening. And I encourage you to look it up, to get involved. We need people involved in this on an active level. So the vision is that you're going to have a partnership for the benefit of society by organizing information. So that's the vision. And the mission is to connect the different data providers in a way that makes sense to make those decisions. So they meet every year, but there's many different elements within GEO that you can participate in. So there's 100 and some countries have not engaged. Don't know why, but this is something that always happens with some of these international things. Now GEO is organized around these societal benefit areas, and there's 8 of them. They make sense. At the top is biodiversity and ecosystem sustainability, which is where we fall in, and the rest of the talk is going to fall under Geo Bond, which is directly related to that first one. But it also has elements on disaster, energy and resource management, food security, transportation, public health, development, and water resources. These are the same topics that permeate all of the different international agreements, like the Sustainable Development Goals, the Aichi targets. All of these have to do with it. So GEO is formulating itself or working so that it can provide the information to address the Sustainable Development Goals. Okay, so then how you organize these different science and technical communities and government communities to come together is not easy. So GEO has organized itself under these. This is a simplistic structure, but you have GEO Flagships, which are the big overarching programs. You have initiatives, and I'll show you a little bit of what those are. And then you have foundational tasks, so the things that actually collect the information that feed into these global programs. The flagships are Geo Bond, so the biodiversity is actually at the top. Whenever you read the Geo implementation plan, biodiversity is the first one that shows up. Then you have these other ones, like agriculture monitoring, you have one on mercury tracking, and you have one on forests. So these, I know you can't read this, but at the top level is the flagships, the four that I mentioned, and then you have the initiatives. So the initiatives are regional organizations. For example, you have AfriGeos, so you have the Group on Earth Observations for Africa. For the Americas, you have AmeriGeos, you have EuroGeos. So there's regional organizations that are trying to then link to these different initiatives. You also have Geo Wetlands. You have Water Watch. And so there's different themes that Geo is trying to address through this process. And the science community, in a way, is the one that is organizing themselves to do this. Then you have the foundational activities. It's like establishing the proper internet networks or broadcasting data to places where you cannot get internet. So these are support activities that GEO has organized. So again, this is GEO with the societal benefit areas, and I'm going to talk about the Biodiversity Observation Network. So the vision of Geo Bond, is the Biodiversity Observation Network, is to organize the global community to collect biodiversity observations in a standardized way to make global maps or country-size maps, so it's effectively maps of biodiversity variables. Okay, so there's a whole discussion going on what is an essential biodiversity variable. And I, again, encourage you to get involved. I'll show you the website to sign up. You can go to these meetings to try to figure out what we mean when we talk about an EBV. So this is Geo Bond. It looks like a bureaucracy, but it's really a lot of people trying to organize themselves around themes. There's a secretariat, and it's now in Leipzig, in Germany. And they have these task forces. The task forces try to organize the different parts of the science community around these activities. So we have the Biodiversity Observation Network, so different bonds. And the bonds can be country-sized, like Colombia has organized a Biodiversity Observation Network at the national scale. But we also have a thematic bond. So we, in the ocean, organized, proposed, got approved this Marine Biodiversity Observation Network, or MBON. So that's where it lives. It lives under these bond structures. There's a bond development task force, and then we have all these different working groups. The working groups are in charge of developing the essential biodiversity variables. So there's a genetic composition working group, species population. So how do you quantify and how do you map species populations? Species traits, which, many of the things that we can measure by satellites are really just traits. The color, the texture, how it changes over time in terms of phenology. Those are traits. And if we can map those over time, we end up with an EBV, okay, with an essential biodiversity variable, and so on. So we have all of these, from ecosystem function to genetics, are EBVs. This is a page. I'm sure you'll have access to this when to these lectures, so I'm not going to stop here. But there's a place where you can register to Geo Bond and then put yourself, well, I'm interested more in terrestrial ecosystems or in regional bonds or in marine bonds. And then you get subscribed to list servers. So in the most essential way, when we talk about essential biodiversity variables, what we're talking about is if you don't have measurements or counts of species, there's no way that you can measure biodiversity. So we go around that in measuring traits because it's easier, or we're measuring habitat diversity because it's easier. But if we don't measure species and count them, then we don't really have a good measure of biodiversity. And that's extremely difficult. Okay? The diversity of life is so large that it's very difficult to do this accurately. But we want to measure the number of species. That's the most basic measure of biodiversity. Abundance in biomass, to me, if I measure carbon, but I don't associate it with numbers of species, then I don't have a measure of biodiversity. And that's what we're doing today. We measure carbon, we measure nitrogen, we measure primary productivity in terms of grams carbon per meter squared per year. That's not a measure of biodiversity. It's a measure of carbon. Okay? It's not a measure of life. Okay? So we need to make sure that once you tie that measurement of carbon to a group of species, then you have an essential biodiversity variable. And I hope that that concept is clear, okay? Because we go around saying that we measure life, but if we don't measure the diversity of life, we're not really measuring, we're measuring one aspect of life. So, but the traditional definition of biodiversity includes ecosystem diversity and habitat diversity. Okay, so this is how people play with these concepts. Okay, so if we want to establish a global network, how do we do that? Okay, so we need to talk to people, we need to convince people that we're of the same mind, we want to do the same thing, and that's not easy at all. Everybody has a different opinion on how things should be done. Everybody has a different opinion and different schedules, so meeting is difficult. If you want to talk to somebody in Africa from the Americas, you know, we're 12 hours apart and 10 hours apart in schedules. Scheduling phone calls is difficult. So all of us involved in trying to organize this spend a lot of time just figuring out when we can meet, and there's way too many meetings, way too much email. And yet, this is what it takes to communicate. Okay? And the bigger the groups, the more complicated it gets. So we try to partition people into how, what, and under what framework we can communicate best for a specific objective. So who are the partners? Why do we need to do this? How? There's an enormous amount of effort going on into simplifying the language on how and why this is important to do so that we can sell the idea to the general public who ultimately pays for all of this, right? So the governments of the world pay for this. So these are the elements that we're trying to put together. We're not ignoring the environment, so we're not just focusing on counting species. We need to understand how those species are related to the environment, so carbon dioxide, temperature, pH, and so on. We do want to find better ways to measure species and how much of one species there is. We want to look at their interactions, so trophic interactions are very, very, very important. We want to do that through proper information systems, so we're organizing the data sets and the metadata protocols so that this can be done in a way that is useful and easy to use by a lot of people. We need to put visualization and modeling into the equation so that we can do species predictions, or what we call species distribution models. So there's a big effort to try to understand how, if I have a certain temperature, pH, oxygen concentration, whether a specific organism will appear someplace. Okay, so those are species distribution models. Or are their ranges shrinking, or are they moving north or south? We need to have engagement with the people that use life. Okay, so fishermen, people that harvest or do tourism. And then we have to have products. If we don't have products, And I think that this is one of the big limitations right now, is what are the products that we can offer people around the world? What can we offer governments around the world? So there's a whole sustainable development goal effort, or Aichi targets that require indicators. And the maturity of indicators is terrible. I mean, there are very few indicators that are worth anything at the moment. They have big, I'll show you some of these later and say, well, how can you use that? Because it says, we'll measure pollution. Okay, well, that's the indicator. How can you measure pollution in a practical way so that it really becomes an actual indicator? So what we're trying to do is come up with ideas for these indicators in terms of life in the sea. And these indicators are of use, hopefully, to inform policy, to come up with policy options or management decisions. So in MBON, we're trying to coordinate between institutions and people, identify resources to, for example, do training courses like we just had here in Brazil a couple weeks ago, and have the capacity building. So this is a diagram that was published by Enrique Pereira. Enrique Pereira is the lead of Geo Bond. He is in Germany right now. But he came up with this diagram and published in a paper in Science. And this was done in 2013. 4 years later, we're still trying to figure out what this diagram means. Okay. There's a lot of discussion about how you measure species traits, especially from satellite or in the field. How do you aggregate it into a map? How do you measure it over time in a way that you can interpret it and then detect change? I mean, what we're really after is detecting change. Change in space, why is that? Or change in time. That's true for genetic composition. The whole field of omics, okay, the genomics, metabolomics, and so on, is very complicated. It changes almost daily. It's not cheap, and a lot of us are looking at it as maybe one solution to a lot of these problems, but there's a lot of issues because everybody does it differently. In one lab, in one place, even in the same state or university, does it differently from the next lab. And so it's very difficult to compare genetic information, let alone doing something across the globe. So if we think that genetic technologies are changing almost weekly, how can we pretend to have a long-term time series of some measure that 40, 50, 100 years from now, we can compare to what happened today? Okay, that's that idea of how we can converge on very simple measures that we can then keep measuring them in a way, even though the technology may change, that still has not permeated the community. We all change the new technology, or we all want that, and then we don't know how to compare to what we did yesterday. So we have these 6 things that are underlined in red are the EBVs, the essential biodiversity variables. So ecosystem function, structure, community composition, species, traits, species populations, which is the most basic one, and then genetic composition. Now, more and more, we're looking at genetic composition as the way to follow species populations, because there's many species that we're identifying that we cannot give a name to a species, genus and species. So we have these, the genetic language about it, but not storing them in things like GenBank in terms of genus and species. And so that's a problem, how we manage biodiversity information. So here we are again. So that was the side of Geo Bond, and we have the essential biodiversity variables. There's the other side of the equation with completely separate government agencies doing the UNESCO side, the UN side. So I'm going to talk a little bit about this side, the left-hand side of this circle, where we have UNESCO and the Intergovernmental Oceanographic Commission at the top. That organizes things like the Global Ocean Teacher Academy. It has the Global Ocean Observing System. It has the Ocean Biodiversity Information System, or OBIS. All of that is under the IOC. And we're trying to link that at the level of MBON so that we can inform GOOS to make the right measurements. And then all of that flows down in the information into this, into the users of information. So the IOC is headquartered at UNESCO in Paris, but they have distributed offices. For example, OBIS is in the Netherlands, in Belgium. But they have a distributed network where many, many different countries around the world have offices of OBIS that link through, and I'll show you a map of that. Goose is headquartered in Paris, so the main office, but yet it's also a distributed entity that has regional alliances. And the whole concept of how Goose is now organized is kind of guided by this document called the Framework for Ocean Observing. The Framework for Ocean Observing was an invention that came together about 10 years ago, actually, yeah, 10 years ago, on something called the Ocean Obs meeting. Every 10 years there's a very, very big meeting that tries to focus on ocean observation. And over time, this meeting has come up with very big ideas, like the Argo floats. The Argo floats go up and down in the ocean to now deeper than 1,000 meters, but for many years, almost 15 to 20 years, these buoys went down to 1,000 meters. They park themselves there, then they come up once a week or whenever you program them. And there's nearly 3,000 of these floats in the ocean right now. We're trying to put new sensors on these buoys, like fluorometers, oxygen sensors, some other things like these plankton cameras. So that concept and the idea to promote different countries to pay for these came out of these Ocean Obs meetings. So the Ocean Obs meeting that came up with FOO, this Framework for Ocean Observing, came up with this diagram. Okay, so it's a very simplistic thing. You define requirements for observations based on need, be it international agreements or user needs for a particular agency in a country. But all of that feeds into this purple box, and that has the technology to collect the observations, be it ships or aircraft or satellites or buoys, whatever. And then you have products. Okay, and there's a feedback loop. If the products are not so good, it doesn't satisfy the requirements then drive the observing system, and then you get new products. So this loop is very important, and we're trying to refine that loop. There's another Ocean Ops meeting in September in Hawaii next year, in 2019, and you should pay attention, participate in the Ocean Ops 19 meeting. There's a website, look it up, Ocean Ops 19. The whole point is to connect users of ocean information to the observing system and improve the observing system to deliver better products. So, and we're trying to come up with an idea of, well, if we have the Framework for Ocean Observing, does it have to mature? Does it have to go to a new level? So Ocean Ops 19 is going to focus on revisiting the FOO and recreate it or improve it. But what we're trying to do is bring users. So if you have ideas and you want to go to Hawaii in September next year, you know, it's not a bad place to go. And maybe you as a network of people that have met here can have a session. Maybe you can organize a session. So I'm on the program committee for Ocean Ops 19. If you have ideas on what sessions we can have in terms of young people networking with each other, let's think about that. So GOOSE is organized, the Global Ocean Observing System is organized in these regional alliances. The regional alliances, there's 15 of these things. Some are very active, some are completely inactive. So the ones that have worked the best, perhaps, are the ones in the US IUS, the Integrated Ocean Observing System, the Europeans with EuroGOOSE, and the Australians with the Integrated Marine Observing System, or IMOS. The rest, you know, they have some participation, some not. And this is an issue because the governments of the region are supposed to be the ones that put the UNESCO has no money. Okay? The IOC has no money to put into this. They just help organize. So the different countries have to take ownership of these regional alliances and make them work. You know, put instruments in the water, generate products. And they all communicate with each other through this framework for ocean observing. But this is what the GOOSE looks like right now. If you were to look around, and there's a website for something called JCOM, JCOM provides this summary. This is what happened in July of this year in the ocean. These are the different observations. So the little dots are drifters, drifting buoys, like the blue dots. There are some, I don't know if you can see this red dot here, but you have some green dots in different places. Especially in the Southern Ocean, there's a lot of little green dots. Those are the biogeochemical Argo floats. So these are these things that go up and down automatically, and they have now oxygen. Most of them are just oxygen. They have a fluorometer, but they're called biogeochemical Argo floats. You have animal tracking. It's a very big deal right now, where we have sensors that get attached to an animal, and you have, it's hard to see in here, but there's a whole effort internationally on tracking animals. You have gliders that have very long routes, and these gliders have, in some cases, very simple measures like temperature, salinity, depth, of course, but some of them have oxygen, some of them have fluorometers, some of them measure light. So you can go on these types of websites and look around. Some of these moorings are permanent, so there's a whole system that is intended to measure the El Nino, and that's these green buoys here in the tropical Pacific. There's something similar in the Atlantic, although it's not as well organized as and not as old as the one in the Pacific. Then we have the satellites. This is just a constellation that NASA has put up for Earth observing. Many of these things, like Terra and Aqua, now Landsats also, Landsat 7 and 8, are used to look at ocean properties. There's some that measure wind, some that measure rain, and a lot of them that measure properties of the atmosphere and land. But the whole point is to try to get the in situ measurements together with the space measurements. And there's other people, other countries around the world that put up satellites. There's also an effort to measure the deep ocean. So this is the deep ocean observing strategy. It's part of GOOSE. The deep ocean is extremely undersampled, and so there's this effort that you can also get involved in, and you should, if you're interested in the stewardship of the deep ocean, called DEWS. Okay? And these are the essential ocean variables. So there's GOOSE has organized different panels, one looking at physics. So these physical variables of the ocean are the same as the essential climate variables, so ECVs. They just got renamed to ocean physics variables. So we have a biogeochemistry, a set of variables, and they're the ones that make sense for dissolving organic carbon, nitrogen, oxygen, and so on. And then we have the biology and ecosystems panel that came up with these, at least preliminary, essential ocean variables for biology. And there's some that are coming up. We have, for example, microbial biomass and diversity is an emerging. We have not defined them yet. And we have a benthic, a broader definition for invertebrates in the bottom. Because there's some benthos here, but a lot of people were complaining that we did not include specific or broader invertebrate communities as an EOV. So we're trying to do that right now. So these panels meet at least once a year. They meet over the phone at least once a month. And so there's a website where we have a definition for each one of these EOVs in terms of what it is, what does it look like, how do you measure it. And so we're trying to have these in a way that is, it provides a platform for a best practice and that can be used by people that are going into the business of ocean observing and make it practical. So some of them are organized by type of animal, some are organized by habitat. In the deep ocean is the same thing. They're trying to copy the model of the EOVs, and there's a series of task teams developing the EOVs for the deep ocean. So get engaged. This is an idea of what it is that is being, most of the things that are measured in the deep ocean are physical variables, okay, temperature, salinity, pressure, very, very, very few biological measurements, and especially very few measurements that are useful to make assessments. So now there's a big push to develop the deep ocean, not only oil and gas, but other types of minerals. So if we don't know what lives down there, there's no way to help industry manage those activities. So this diagram here is we're trying to make this link between essential ocean variables and essential biodiversity variables. I think that you have access to a paper that describes this, what we thought was a good way to link them. And we came up with this spaghetti diagram that is almost impossible to read from where you are, but we're trying to make sure that people understand that these different EOVs, the biological EOVs, actually can be linked to EBVs. So even though they're developed by completely different groups, that there is really is almost the same. Okay, so the promise that we're starting to duplicate efforts, and we need to bring the communities together that look at these EBVs in a way that is compatible with the EOVs. Then we're also trying to make these mind maps. Okay, these are Sankey diagrams. So in this case, it was one for coral bleaching, and it's a map. If a government entity wants to look at, well, what do I do if I want to measure or understand coral bleaching, we're going to put these things on a website where you can click on the different problems or issues. In this case, it's bleaching over here, and it's related to ocean health, but it's also related to climate. But then you can see which programs, ultimately, which observing network and which of the international coordinating networks manages the different measurements that are relevant to coral bleaching. Okay, so many people are interested in the bureaucracy. Some people are interested in the actual measurement, and this is how this relates to coral reef cover or coral bleaching. Okay, so we're trying to do that for each one of the EOVs. And there's the same framework for ocean observing diagram. In this case, we have the requirements that are set by GOOS. We have MBON trying to define the type of measurements that we need to understand life in the ocean. And then we're working with a other partner, which is OBIS, the Ocean Biogeographic Information System, which is an open data sharing platform. And yesterday somebody asked, well, if we knew of any platform that allowed you to share data openly. There's several, and GBIF is one, the Global Biodiversity Information Facility, and which is general for terrestrial and oceanic, and then there's OBIS, which is really focused on the ocean. The problem with OBIS is that it was created under something called the Census of Marine Life. Census of Marine Life stopped. It was a 10-year effort funded privately, and when that money ended, a lot of the data that was flowing into OBIS stopped. So we need to find ways to encourage people to put data into OBIS in a continuing way, in a way that you can make assessments of life in the sea on a continuing basis. So what we want to do, and I'll show you what our data sets in the ocean look like right now. This is if you were to look at OBIS, our best, 50 million records of life, of biodiversity, since the 1700s. And if you look at the records in the upper 20 meters of the ocean, this is what you get, a lot of holes. You have some areas that are well covered near the coast or near industrialized nations, but most of the ocean, we don't have even a single record in the surface ocean in OBIS. So that's not good. This is the way that it's distributed with latitude, and this is the way it's distributed with time. So if you look at the last 10 years, there's a big decrease in the amount of data that's being put into OBIS. So here we are saying that biodiversity is being lost, that we're making assessments with IPBES, that we're going to make assessments of the global ocean because life is changing. And we cannot do that. Realistically, we cannot do that because our best data in OBIS to do this stopped being, it's not completely stopped, but if you think that we can make assessments of change with a decreasing data set, it's not realistic. Okay? So we need to find a way to have information that is continuous and sustained. So a lot of the effort that we're trying to do is do that with tools like satellite data sets, with traditional data sets. So we have we're trying to organize people that have existing monitoring sites, like time series stations. And there's some that come and go. For example, we had one off Venezuela that we called the Cariaco Time Series Station, and then we lost the money. We measured for 22 years, and then the money went away. So some of these things can take a lot of effort, and then all of a sudden, if you lose the money, it's gone. And yet there's other ones that come up in other places. And one lesson that you should probably learn is don't start a time series if you can't keep it up. Because it's a lot of effort, and then it's a disaster when you lose it. But typically, they do have a finite lifetime. We're trying to combine traditional measures of biodiversity, be it species counts or pigment concentration or genetics, with satellite data and something that we call seascapes. And I'll talk a little bit about how we come together with these seascapes. Seascapes are an artificial way for us to come up with biogeographic areas, with the idea that maybe that will inform us about where things live. So we're trying to come up with, again, the idea of MBON as a network, so we're trying to come up with long-term ecological research sites, link the GOAON, the Global Ocean Acidification Network, things like OBIS and GOOSE, and serve the broadest community possible. So traditionally, we measure diversity in the ocean with these things. We have species richness, Simpson index, you know, the different people like Margalef came up with indices. We have all sorts of indices that are hard to implement in a way that makes sense. Okay, so if I go to a database and today I sample bird, maybe 2 or 3 kinds of birds, and 4 mammals, and 5 phytoplankton, and put it into a formula, I get a number. Okay? And so people are doing that, and they say, well, these are the data available for an area, and I'm going to mix microbial communities and fish and birds and some turtles, and I'm going to call it a biodiversity number. Well, that's nonsense. Okay? This is a very dangerous thing that a lot of people do, and you should not do that. Okay? You should compare turtles with turtles and fish with fish and bacteria with bacteria, and yet people don't do that. People take whatever is available and throw it into a biodiversity index, and then they say, oh, I'm going to do that 5 years from now. And they measure another group of phytoplankton, another type of bird, and another type of fish, and then they compare it and they say, oh, it changed, you know, biodiversity changed, or maybe it didn't change. But it makes absolutely no sense the way that we're managing biodiversity data. You need to be very careful about that. So what we're recommending is that you stay within a certain trophic level and compare at least species information at that level, you know? Be it microbial or primary producers or top-level organisms in the food chain. The other way to do this, in which you do mix trophic levels, is through functional diversity. So functional diversity is where you take things, well, all the things that consume nitrogen or that produce carbon, like primary producers, or all the fish that are a certain size or have a certain habitat. Well, you can compare the diversity within those groups or across groups in a way that is a function in the environment or in the ecosystem. But measuring species and then throwing them all in a bag, you're never going to have all the species to make a real robust index. So be very careful about that. So what we're trying to do with MBON is take databases and data sets and be able to filter them through taxa, through space and time, and then come up with maps that can tell us something, some indicator of abundance, and then how it changes over time. So status, this is trends. Okay, so these are the indicators that we're going to be after. Now, how much time do I have? 15. Okay. And to do that, MBON is trying to come up with mechanisms to integrate and visualize different data sets. So we have, we take these seascapes done by satellite. We take things like something we call environmental DNA or eDNA, and I'll talk a little bit about that. And we take other environmental data and we try to smash it all together into a visualization. For that, we're taking satellite data, in situ data, genetic data, the biodiversity data, and one thing that we're still missing is understanding how we bring in socioeconomic data. All of these data are important in an observing system, and we don't really know how to do this, how to bring all of these things together in a useful way, in an operational way. One of the things that we're trying to do is do a little bit of data archaeology, and that's something that I would encourage you to do. There's a lot of data that sits in people's computers or in an agency archive that is not publicly available, and eventually those data are lost. If you have an investigator in a university that doesn't publish their data, doesn't share it, it's like that data never existed, and eventually that will be lost. Okay, because once that person becomes a taxi driver or dies, that's the end of that. We're trying to bring in the data under what GBIF and OBIS use as a standard, which is Darwin Core. Darwin Core is a schema for data. It's basically a table with specific names of columns. That's what we call these data set cores. And Darwin Core is one that is becoming very popular. And it's changing all the time, so we need to keep up with what Darwin Core is. But we also want to bring in environmental data set into these layers. We're trying to bring in bioptical data, so that tells us something about the physiology of especially primary producers, something about water quality, satellite data, and all of these other things. So in terms of seascapes, many of you may be familiar with this paper by Alan Longhurst. Longhurst came up with the idea that the ocean really can be somewhat predictable in the way that you can have environments where things live in, and he called them provinces. So these are biogeographic provinces. So he had these very large, like polar and some in the mid-latitudes, a tropical for the trade winds. And within each one of those, he had specific, so these are big biomes, like the polar biome or the trade wind biome. And within each one of those, you had provinces. But if you look at these, they don't change over time. They don't even change over a season. So we know that this is wrong. We know that the ocean changes with seasons, and yet how do you capture that? Okay, so how do you do that? And that's where we're coming in with the seascapes, where we take now satellite data that is global, like light, color, temperature, wind, altimetry. There's many different variables that you can put into something like this, and combine them into the same construct. Because you have a classification, it's a thematic classification of these different variables, but they change over time, not only over the seasons, but from year to year. They're very dynamic. And so that's what we call seascapes. Problem with the satellite data is what? Give me one problem. There's many problems with satellite data, but give me one. Clouds, yeah, of course clouds. We don't even want to talk about clouds. Clouds affect some types of satellite data, like visible, infrared, but it doesn't affect radar or microwave. So you can still measure temperature or salinity with microwaves through clouds, but you cannot measure the color, because in 50% of the world, at any one point, is covered by clouds. So you're obscured. But if you take measurements every day, frequently, then you can fill the holes. So I'm not too worried about clouds and the kind of thing that we're doing, because we're using weekly means or monthly means. What's another really serious, probably one of the most serious issues with satellite data? What? Well, resolution, yeah, but again, at the global level, not too bad. We have satellites that measure things at a quarter degree. Many measure at, nowadays at a few centimeters. So I'm not too worried about resolution, and for what we're doing, it's not the issue. Somebody said something?
I said it's expensive.
No, it's not expensive. Nowadays, for this, we can, okay, somebody pays for it. But, for the amount of data you get, each data point is very cheap. If you go out on a ship and you collect a bottle of water, that is expensive. That sample can be $10,000 each little bottle of water. So it's not, you only see the surface. Okay, we miss kilometers of ocean habitat by only using satellite data. So satellites have that little limitation. Okay, just be very, very aware of that. So the biogeographic areas that we're coming up with are only the surface. This is a complicated diagram, but we make these classifications. We try to see if they're real. We go out on a ship, or we have gliders. We compare it with real information. We refine the classification, and we try to see if it's useful for anything, to make a decision or to understand the environment. So these are examples of what we're doing. This is how we started. We started doing these off of smaller regions where we had field programs. And we're now trying to work with people around the world that have other field programs. So this is Florida, this is Cuba here, the Bahamas over here. This is California, and this area here is this is Monterey Bay. So this is the Pacific, the Atlantic. And we see colors changing. So these patterns are real. In this case, we're trying to combine a classification that includes chlorophyll, fluorescence, measured by satellite, temperature, and light. But we're now bringing in other things. No? In this case, we use the seascapes to guide the cruises that we make. So these little stars are samples that we have collected over the past 4 years within different types of seascapes to try to understand what lives in there. So we measure phytoplankton, bacteria, we collect eDNA samples. In some cases, we have fish data for the Florida Keys. And we're trying to do this now at a global level. So we started with small regions, and we're now doing this at a global level in an operational way. So we're now going through the tests. We're trying to understand what they mean, what do the different colors mean. For example, off of California, we compared, for example, to anchovy concentrations. And then we can, depending on what type of seascape you have, this is just a map of one specific seascape. You can see if there is habitat for anchovy or not, or habitat for whales, or habitats for crabs. So, for example, off of Florida, we have we're doing something very simple. We took samples of phytoplankton in bottles, and we count them through a microscope, and then we can see, well, these are diatoms. We have things like haptophytes, like coccolithophores, in some areas, and we see that there, in fact, yes, there are spatial patterns that coincide with the seascapes, with more diatoms as the more closer you get to the coast. So some of these patterns are real. They're very hard to interpret, and they're extremely hard to interpret on a global level because a certain seascape that you may have in the polar areas may be something very different from what you have in an upwelling area. So we're trying to go through the process of documenting that right now. We're also comparing seascapes to fisheries landings. So we took the seascapes that we were producing and compared it to fisheries landings data and this project that Dan Pauly has called The Sea Around Us. So we took all of the fisheries data and these things called large marine ecosystems of the Americas. And we did a classification where we did just the physical variables, and then we also added the diversity of fisheries and compared it to the diversity of seascapes. And so we ended up identifying things that, you know, I don't know how many of you know about these large marine ecosystems. It's the same thing that Longhurst did. It's a geographic classification based on some biological and physical parameters, but they're static. Their boundaries don't change. Okay, so each one of these boundaries should change with the seasons or with time, but the LMEs don't change. So what we thought, well, let's test the hypothesis that in fact some of the LMEs are larger than what is proposed. And so we called them mega regions, and we found that if we combine fisheries with, which is really the whole driver behind LMEs, is that you do have LMEs that are larger than just one LME. And that's, you know, that's because of landings, okay? That's driven by the species that are landed, but there's also things like regulations that come across over time, and when regulations change, that may change the way that you define an LME. So anyway, this is just playing games with what we can do with seascapes. One of the purposes of seascapes is trying to understand functional diversity. So one of the things that we are trying to understand in the ocean is whether different types of phytoplankton give a different signal that we can measure by satellite, and then we can call them functional groups. phytoplankton functional groups. And so you have things like nitrogen fixers, that's one functional group. You may have, divide them by size, like nanoplankton, picoplankton, or microplankton. Those would be other types of functional groups. And the reason for that is, the utility of that is that different functional groups may lead to different types of food chains. Okay, so you have, in coastal areas, you have diatoms, which are bigger cells, and they may lead to a certain type of food chain in fish. And on the open ocean, where the cells are smaller, the phytoplankton tend to be smaller. You may have a longer food chain that deals, but you end up with different kinds of fish. So if we can characterize the ocean in simple ways with these biogeographic regions or seascapes, we may be able to say something about how these food chains are changing. And we know that the ocean is changing because we know that the wind is changing, we know that the temperature is changing, and we know that chlorophyll patterns are changing, not only seasonally, but over the long term. So what's the impact of those changes on these food chains? What's the impact on carbon flux to the bottom? Okay, so if you have big cells relative to small cells, we may be able to say something about how much carbon gets taken away from the surface ocean to the bottom. These are experiments that are being done. There's a guy called David Siegel, but there's other people doing this. They're trying to take satellite data and estimate how much phytoplankton there is in different size classes. So you have the small nanoplankton or picoplankton and microplankton. These are the bigger ones. You see that the bigger cells are all mainly in the temperate latitudes, or where you have upwelling, or in coastal zones. And yet you have the smallest cells all over the place. And the same thing with the volume, the biovolume of these cells. So microplankton is, again, most of the biomass. It's not just abundance, but the biomass is concentrated in temperate areas, upwelling areas, and coastal zones, as opposed to the nanoplankton and picoplankton, which is really more in the middle of the ocean. And these patterns make sense. I don't know if this is a good place to stop, so we can just take a break, and I'll continue with what we call how can you measure essential biodiversity variables from space, which is another thing that we're trying to push.